right, thank you very much. Again, for those of you who might not have been here at the beginning of last presentation, my name is Jenna Ridlen. I'm with the biology department. Thank you so much for being here. This is BriAnn Buford. So whereas our last two scientists have been researching with me for all last semester and all this time so far, BriAnn really has had her project started maybe about 2 weeks ago.
That's generous.
And has done a wonderful job catching up in the process, and with an advisor who's intermittently available, and is also going to be presenting at the AAS conference in March. And so hers is a PowerPoint presentation instead of a poster, and she has some personal connections with lymphoma, which is why she decided to choose a gene that was relevant to lymphoma. So BriAnn Buford, one of our rockstar scientists in the biology department.
Hi, well, as Dr. Ridlen introduced, my name is BriAnn Buford. I am a senior this year, and I decided this semester to dive into some research, and she kind of passed the ball back to me and said, okay, I know you're interested in like genomic research, I know you're interested in genetics, but like, what do you have a passion about that's going to help you to pursue a project like this? And in April of 1990, my mom was diagnosed with classical Hodgkin lymphoma. It was stage 3 at the time. I still have the picture to this day that my parents had to take because they were convinced that she was going to die from it. What we consider to be common treatment now was actually experimental at the time. So as I was asking my mom questions about this, trying to learn more, I was met with I don't know, actually. They never actually explained that one, or it was new at the time, or they didn't classify it that way then. So I do have a personal connection to this story that I'm going to try and tell, and my goal is to understand it more than even I'm telling you today. So if you have a program or like information about the title, you'll notice something has changed a little bit in the title. I actually selected a gene that was directly connected to Hodgkin's lymphoma, or the phenotypic expression of it. But there's so little research on Hodgkin's lymphoma that we actually couldn't use that because there was not enough raw data to actually even start. So that's partially why I decided to do this, because I have a passion to learn more, and hopefully those data points won't be so rare. But this is gene variant TNFAIP. You'll find later that it is tumor necrosis factor alpha-induced protein 3. And I'm going to try, and in the most understandable way, make sense of something that makes my head spin daily. Hodgkin lymphoma, if you don't know, is an infection of the lymphatic system. Basically, it means that there's an overgrowth of white blood cells and lymphocytes, hence the name lymphoma. Most people who are infected with this disease have something called a Reed-Sternberg cell. You get pretty typical symptoms: fever, aches, chills, but it's commonly associated with swelling of the lymph nodes. This can be painless or painful, so that can be anywhere from the neck to the armpits. If you were my mother, you were diagnosed with congestion and a stomach bug, and that's what they diagnosed her with before they found an actual tumor over her heart. So they have predicted many different genes associated with this, but the cause is still considered scientifically to be unknown. Okay, so these are two main players in this kind of game that lymphoma is playing physiologically. This is the gene that we're talking about, TNFAIP3, but NFKB, or the nuclear factor KB, is actually the pathway that it is affecting. So this gene acts as a regulatory product of this pathway. So it's an important factor, and it plays a role in inflammation and immunity responses. So when your cells are sending out signals saying something's wrong, I need to stimulate a response, this is the pathway directly involved in it. Why does that matter? Well, this protein controls that, or helps control it. So this is a nice little portrait of the gene that we're talking about. Like some of my actual colleagues back there were mentioning, PDB files are really hard to find, especially about things that are under research. So I used Visara as the software as well to visualize this. However, it took several days to find a PDB file because it's so under research. Most of them were too short, some of them had misalignments in the nucleotide sequence and the amino acid sequence. So we had to do some serious digging, but this is our gene in question. Once I was able to find this gene in ClinVar, I was able to lay out all of our data points that we have, which is what you see in the graph in front of you. Anything you see in blue is considered benign. Anything you see in red, which is 1 point, is pathogenic. And then anything in green is uncertain. These are all the data points associated. I didn't take any out, I didn't add any. So this is all we have to go off of for this so far. So we only have one known pathogenic variation, and then we have a pretty substantial percentage of unknowns. Basically, it's by residue number and by CAD score. CAD score, as my colleague so wonderfully explained, is a prediction of pathogenicity. It's not a guarantee, because as we learn, just because it's higher doesn't necessarily mean it's pathogenic, and just because it's lower doesn't necessarily mean it's benign. But it is predicting the deleteriousness of a protein. So when I went about this, I looked at all of the data points that we had as unknown and decided which one seemed beneficial to me to investigate to see if it would actually cause a connection to phenotypic expression of Hodgkin lymphoma. So I chose a residue 558. I could point it out to you on the diagram, however you'd have to squint really hard to see it. It's close to the end of our sequence. Our sequence has over 780 residues attached to it, and this is actually the change that you see occur. I picked this particular residue because if you look at the raw data points laid out for you, this was the closest residue to the CAD score of the pathogenic variant. So there's a couple of different ways I could have gone about picking it, and that was what I chose to do, to say, hey, if it looks close to it in raw data, will it actually be close to it in expression? And so what happens at residue 758 is you have a proline residue that is changed to a leucine residue. Proline and leucine, obviously, part of the 20 amino acids that make up all of our genes. All of them are significant. Proline has a major role in structure, and leucine does a lot of activity in metabolism. So this is actually the simulated versions of those. So you can see proline in the original as that green that we associate with it, and then you see leucine altered. So you do see a structural change. It's in magenta. It's also labeled for you. And so this is the change that's occurring. And so we know that something is causing this to switch and affecting the pathway that keeps us from having inflammation responses. So the story is short, but it's enough to dig at. This is the polyphen software. And so after investigating what the change would look like, I actually plugged this gene and this switch into polyphen to determine what the outcome would be predicted to be, as well as to see how conserved this gene is across various species. And so naturally it predicted it to be likely damaging, as well as you can see this proline that's highlighted right here is the residue in question. It is highly conserved across many species. So we know from the story so far, a change occurs, it makes a structural difference, and for some reason, it's connecting itself closely to the pathogenic variant. All this to tell us that the odds of this producing a pathogenic response are really high. What does this tell us for future? Well, one, it tells me that I have more to investigate. 2 weeks of research has led me this far, so I can't imagine what 2 more weeks can get me. So I imagine that I get to have a lot more fun investigating this and digging into this and learning more about it and learning more about the personal connection that I have to it. But also this could be a potential target for drug therapy one day. This could answer some of the questions about what actually is causing this disease. Is it something genetic? Is it something mutated? We don't know. And so this is adding more to the puzzle pieces every day of what causes this disease. And hopefully the goal one day is to have so much information that we can conquer it. Thank you.
Any question for the speaker? Yeah.
Yeah. First of all, thank you for presenting. 2 weeks is nothing for research.
I know.
But you started really nicely. Can you please go back the data catch code?
Yes. Do you want to see the graph?
I know the dot scattered.
The scatter plot.
So
Yes.
here mostly we see the green dots, right?
Mm-hmm.
So like, what does it mean? Like what's the,
So this is going off of like what we know so far versus what we don't know. So this is kind of trying to give you a visualization of where it is along the molecule compared to how it is predicted to be pathogenic. So it's kind of like a really reversed map. That's how I would put it. That's how I use it as. So anything you see in blue, they know it is benign. So you know anything in blue on this residue number with this CAD score is going to be considered benign.
red one we see only one.
Yes, there's only one known pathogenic mutation. So as far as the data has been tested, we only have clinical evidence that one of them will be pathogenic. And it's a pretty big one. It's actually a mutation of a cysteine residue, which means that disulfide bond that causes a lot of important regulatory factors in terms of structure. That is what it is.
Any more questions? Yeah.
So you had picked 758 residue
Yes.
because it had a similar CAD score to your pathogenic.
Yes.
If I'm looking correctly, this is my question, am I interpreting it correctly? If you go to the left at about 150 residue, there's a blue dot that says that's very likely benign, and it is also very close to that same CAD score, correct?
It is. Yes.
I'm just wondering if we could do a comparison between the benign and the pathogenic with the one that you're looking at to see where it kind of falls. Does that make sense?
Absolutely. Absolutely. And you know what? I'm gonna have to invite you the next time I present this. so that it's a good thing, so that you'll see it when I put it on my PowerPoint. Thank you, I appreciate that. Yes, that would be a fantastic measure to have on here.
More questions or comments? All right, if no more questions, let us thank our speaker again.
AlphaFold's a Google company. They won like the poster bioinformatics contest in 2022, and so they're like, just like brain technology or whatever. Sometimes it works. Maybe we'll get you.